Hello, welcome to my presentation for bioinformatics. My name is Patrick O'Mara, and I'll just click onto this. And basically, my project was aligning proteins using a amino acid similarity matrix derived from MHC peptide binding for use in antigen. Right into it, kind of explain exactly what my project was all about. So first of all, I just wanted to get it in, just go through an outline real quick of what I'm going to talk about. First, I wanted to just talk about quickly about what is antigen design, why is it important, involved in antigen design, and what. And so also, I also wanted to get into why similarity, why matrices are important, why protein alignments could be important for antigen design. Next, I'll get into protein alignments a little bit, explain some of that, and what I use to do these alignments, why I chose that. Next, I'll go through the analysis of and some of the results that I got from these alignments, and what kind of conclusions I came up with, and obviously what to do in the future. So going right into it. The first thing is just briefly explaining what are antibodies and antigens. Well, antibodies are, in molecular biology, there's different, usually scientists are trying to figure out ways to locate and quantify proteins. Now, there's various ways you can do that. Markers proven to be a valuable tool in molecular biology, as they don't interfere necessarily with the function of the protein itself, or they don't do it as much as maybe a tag or something would. So in this sense, they have become a vital part of molecular biology in the last few decades. So basically, and what is an antigen? An antigen is the part of the antibody, is the part of the protein that the antibody binds to. Now, so in essence, in antigen design, what we're trying to do is we're trying to find good antigens and good regions on proteins that an antibody could bind to. And what makes a good antibody? I mean, what makes a good antigen? Well, first of all, it has to be specific to the protein itself, right? Because if it's not specific, then an antibody could bind to a few different types of proteins, and then you don't know exactly that you even are tagging this protein. The antibody is not going to bind to it, and that's pretty straightforward. And meaning that the antibody has to be able to bind to it in physical space. For various applications, this is important. For applications like Western blot, this is not as important as the protein's denatured, but applications like IHC or IF, this is very important because the protein could actually be folded and the antigen could be buried within the protein itself and not accessible. So first of all, we have to consider, how do we find these, how do we determine regions that are good for antigen design? Well, the first step in this process is actually performing protein alignment. Well, we perform protein alignments on the target protein because they provide us with regions that might be specific mostly to our target of choice. So for our purposes, usually what you can align any kind of protein to your target protein depending on what you want. You can they're similar, if you actually want cross-reactivity between different species. But the main alignment in this case is actually you want to find the nearest protein to it, and you want to find those dissimilar regions between the two proteins. So in this case, we're going to find the closest protein and closest manually annotated protein, or from the Swiss Pro database. We're going to perform an alignment between the two. And we're going to find the regions that are not similar. So this is just an example of what this might look like. Now, this is just a region of the alignment. The protein is much larger than this, but I want to show elements of what good antigen design looks like. So this protein right here, there's an area in red, which is actually a real antigen. It's published in a paper. So we have to ask ourselves these three questions. Is this specific? Well, there's 4 acids similar between the two alignments. But as we can see, and for an epitope, in which antigens, epitopes in them, the epitope is the actual region where the antibody binds to, but I don't want to get too much into those details. But basically, an epitope is around 4 or 5 amino acids, in length typically. So, you might ask yourself, wow, that stretch right there, I mean, so they could bind to that. But if you look around it, you can see that there's a glutamine, and a lysine aligning across from each other on one side of it, and the other side there's an alanine and a phenylalanine aligning on one side. On the other side, those amino acids are very different. The glutamine and the lysine, very different. One's positive, one's not. On the other side, the phenylalanine is hydrophobic, it's large, whereas alanine is, is very small. So in this case, if we look at it as a whole, we see that it's actually very specific to the protein choice. Now you can do other types of analysis to see specificity, which I won't get into detail, but for our purposes, this is very specific. We look at if it's immunogenic, and there's been much about immunogenicity of amino acids, but namely glutamine, anything that's charged, so lysine and the glutamic acid, they're considered always to be immunogenic. Proline is a good one too, because it kind of anchors the antibody in place, and phenylalanine and tyrosine are actually immunogenic as well, even though they're fairly hydrophobic somewhat, at least phenylalanine is. They're large, and they kind of get this immunogenic response. So it's immunogenic, so that's good. And is it accessible? Well, for Western blot, it obviously is, but for other applications, I looked. Actually, there was structure available in this region as well, and it was actually fairly open. I don't know if the paper that used this actually used the antibody for other applications, such as IHC or IF or something like that. But it's actually, it's a fairly good antigen, and obviously it works. There's a antibody made for it, so it kind of highlights in this kind of we're looking for in good antigen design. So, anyways, when what is the typical substitution matrix the alignments are derived from? Well, usually their matrix is derived from point mutations or some kind of evolutionary standpoint. Overall, these are great. I can't complain too much about them. There's one concern that I was interested in, is kind of the focus of this whole project is, first of all, mutation rates are higher in loop regions, right? So loop regions are these regions that are unstructured. they usually don't have much function. They usually are kind of like hinges, or they allow the protein to be more solvent accessible, or provide some kind of flexibility. But they don't really have as much function as some of the other regions. So what we find is there's more higher mutation rate in these loop regions. Now, since loops are usually more solvent accessible, they're usually hydrophilic amino acids include more charge residues, so charged amino acids. So what happens in these evolutionary is amino acids that are oppositely charged get more, they're more similar than probably what their physiochemical properties would suggest. Mainly because, if you change a positive to a negatively charged amino acid in a loop, it's really not going to affect the protein all that much. Or it's perform that type of thing for the protein as the other, as it would do for the wild type protein, the non-mutant protein. So this was something I was interested in, and I wanted to basically I wanted to investigate this, and I actually found this paper. I thought it was interesting, and they kind of talked about this, and they actually made a matrix, a amino acid similarity matrix based off of a peptide and how a peptide and an MHC molecule bind together. And so basically these amino acids are based off, or the similarity between them is measured by how they bind. So I thought this was very interesting, and I wanted to see if I could apply this to put these in use this as the basis for alignment, protein alignment, and see what I could do for antigen design purposes. So so the matrix and muscle. So basically, in order to do this, I had to look up programs in order to do this. And muscle actually provides this flexibility. They allow you to use your own custom matrix. And also muscle, a well-known program, a lot of literature, has suggested it's one of the better ones out there. So I felt very comfortable using it. So I downloaded that, and I've used that. Quickly, the full 240. There are some papers talking about this, but unfortunately, I could not find anything. I could not find the gap penalties in any paper. I could not find the default muscle alignments actually use, which was unfortunate. However, in a paper, I found that the VTML 200 matrix is actually very similar to the default. And in that paper, they actually had optimized gap penalty. So what I did was I actually compared the MAC matrix to these two matrices, and I got very different results in the end. And I'll get more into detail about that as we go through. So first, to look for proteins to align. So what I did was I looked for 10 proteins on IEDB. It's just a database full of epitopes and whatnot, with known linear epitopes. I didn't want discontinuous epitopes, I wanted linear, 8 to 14 amino acids in length. So I found those, and I blasted the 10, and I found the closest protein. I had to drop two I dropped two basically because those two proteins actually, the most close similar protein was actually not all that close. And for our purposes, if it's not that close, then there's nothing really to compare it to. And then, what's the point? So I dropped those two, and So we were left with 8. Performed 3 alignments on each target protein comparison protein pair. And I ended up with basically 24 alignments total. So we'll look at the, so anyways, so we'll just dive into the analysis. So basically, sorry about that. There we go. So anyways, sorry about that. I wanted to dive right into the analysis. So basically, first of all, I wanted to analyze the protein as a whole. And to do that, I wanted to look at certain different parameters. And what I did was I actually looked at each position in the alignment, the percentage of those positions. And the things that I measured was the percentage of positions in the alignment that had gaps, that had oppositely charged amino acids, that had the same charge and also the same amino acid. For our purposes, I really only looked at the gaps in the oppositely charged. And what did I really do with those 2 parameters? Well, I wanted the 2 alignments, so the MHC and then one of the VTML, which one I was comparing it to. I wanted the percentage of gaps to be similar, and I also wanted it to be low. In answer to design, you can't really have alignments with huge gaps. It doesn't really make any sense. You know, you're looking at these things, there's huge gaps in between. You kind of want everything to be restricted and small. But you definitely want them to be similar. So I just use an inference test to figure that out, a z-test, to figure out that property. And then if they are similar, if they are significantly similar, you know, the hope is that the percentage of oppositely charged amino acids would be derived alignments. And I did a one-tailed t-test because I'm trying to figure out if one's better than the other. So anyways, here is the data for the AMHC versus the VTML 200. So this is not the default. This is just the one I found off that paper. And as you can see, so just real quickly, we can go through and we can see that the green bars are the equally charged, or sorry, not equally charged. They're equal amino acids, so basically they're the same amino acids in the group. The purple is the same charge, but like once again, I'm not going to get too much into that in this presentation. The is in red, and the gaps are in blue. See, 6 of the 8, and these are the z-scores for them. 6 of the 8 proteins has significantly there's a significant difference between the MHC and the VTML 200. Basically, what this shows is that there was a lot of 100 alignment versus the MHC alignment using the parameters set in the paper, described in the paper. So, and if we just keep on going, there's only one that showed that the MHC, let's see, yeah, there was any significant difference in the oppositely charged residues. Basically, this was not good, right? Because we wanted the amount of gaps to be similar. That was kind of one of the parameters we set beforehand. And there was only one protein that showed that there was there was any kind of gain or there was less, sorry, there was only one protein where the amount of oppositely charged positions was less in the MHC protein. But by far the largest for the VTML, it was impossible to really compare the two. So unfortunately, I had to, due to the MHC derived was better. Due to the high number of gaps in the VTML 200, I concluded that the MA derived alignments were actually better, because that was kind of one of the parameters we set. But we couldn't really conclude whether or not it really gave better oppositely charged results. But if we start to look at the other one, and I found this one was a lot more interesting to me. So this is the MHC derived alignments compared to the VTML 240 or the default alignments. These are the z-scores. And as you can see, there are two proteins where the number of gaps are significantly different. The MHC actually had a higher number of gaps than the default. Now, interestingly enough, these two proteins that this occurred, these two were the comparison protein for these two was the least similar out of the 8. So basically, out of the 8 proteins, when I did the blast search, these two proteins had the lowest identity percentage out of to their comparison protein out of all the 8. So naturally, I think it's easy to conclude that if you have a comparison protein that's so, you're going to get two very different types of alignments, and therefore I think that is reflected in the number of gaps that are present. Now, we look at the difference, see if there's any significant difference in the amount of position or the percentage of positions that have oppositely charged amino acids. And as we can see, there's two, and it's the same protein. So basically, there are less, and this is showing that there's significantly less positions with oppositely charged versus the default alignment. However, as we stated before, we needed them to be about the same in order to really conclude that what we're seeing here is an effort to kind of move the two amino acids out from each other and not align them with each other, instead of this just widespread kind of gap and kind of dissimilar alignments across the board. So unfortunately, I had to conclude, or not even so much unfortunately, maybe not so much even unfortunately, but basically I concluded that the alignments actually gave very similar results. But I couldn't conclude that the MHC one was better. So basically I concluded that they were the same for the most part, but they were just couldn't, the MHC wasn't any better than the other one. The next step of analysis was I actually analyzed the epitope regions themselves. Now, because there's only 8 proteins, there's only 8 epitopes, so the sample number is pretty low, and the number of epitopes, the number of amino acids in those epitopes is low. So to do any kind of real statistical analysis, it was pretty hard. But what I did was I just counted the number of positions that in the epitopes that had oppositely charged amino acids in each alignment. And I wanted to see if there was a difference. So I actually start with the MHC versus the default here. what I found was 6 of the 8 proteins actually had the same alignment at the epitope region. So that means that they were, you know, the MHC versus the VTML200 or the 240, the default, actually had the same alignments at the epitope region. So that kind of reinforced my idea that these two different matrices were, basically making the same alignments. So, and if we look at the other 2 proteins where they were different, we will find that one of the proteins had, for the MHC alignment, had an oppositely charged amino acid, two oppositely charged amino acids present in the alignment together. But in the other protein, it came in the. So what we see is we kind of, if we're trying to see if one has, if there's more in oppositely charged amino acids in one of the epitopes and the other, but what happens here is they kind of cancel each other out. So in essence, this also kind of reinforced my idea that they were similar, and that I could not conclude that that the MHC was better or was not, was better or not. That was inconclusive. I think that it was interesting that looking at such a small scale, that this kind of result, it kind of came to the same results in the end. And last, I looked at the VTML200. 2 of the 8 proteins had the exact same alignment at the epitope region. 3 of the 8 differed by 3 positions or less. 3 positions is actually quite a bit when you're talking about such a small region, but they were, because the VTML200 had so many gaps, it was actually gaps, if I recall, especially for the one that differed. So it wasn't as bad as it might seem. However, so basically what it says here is that there actually were fairly similar. I mean, 5 out of 8, you got something that's, these alignments are fairly similar in the epitope region. Interestingly, no epitope region in the VTML200 derived alignments had any oppositely charged amino acids present. Now, and that you might think that's a good thing, right? That's what you're after. However, in about half of these alignments in these epitope regions, the number of gaps was very, very large, and kind of disrupted the whole epitope and disrupted the whole alignment itself. So in the end, I really couldn't determine if it was, I couldn't say it was better because the number of gaps was so high. So once again, this kind of reinforced what I had previously derived or concluded in the analysis, that you couldn't say that the MHC was better than the VTM or vice versa. And in fact, I think what happened here, what it really concluded was that it was just such a different alignments because of the number of gaps introduced into the VTML200 alignments, that it was hard to compare. So conclusions, kind of reiterate what I had said before. I could not conclude that the MHC matrix was better than either of the other 2 matrices based on my parameters. I think at best what I could say is that the MHC matrix basically made alignments similar to the VTML40, so the default, and maybe slightly better than the VTML200, basically because of the large number of gaps. And overall, I was actually fairly impressed by the MHC produced results because they were so close to the default. Now, when I embarked on this, I did not know if this was even going to work. So plugging it in, plugging in this matrix, and seeing these types of results that are so close to what muscle gets, I was actually fairly impressed by that. So just few considerations. I mean, there's a lot that I would love to do with this if I had time and if I had some time. But the main things are basically I wanted to. It'd be nice to be able to compare this MHC matrix to matrices derived from the physiochemical properties of amino acids. I also wanted to see if there's any other ways we can measure if an alignment is better for antigen design or not. And also, we looked at charged amino acids, but is there a way to factor in other amino acid properties such as hydrophobicity or size as a parameter into things? Stated here is that the VTML 200 alignment, even though the gap penalties are optimized in the paper, could I have optimized them myself for antigen design purposes? That is something that I would like to do in the future if I had some time. But yeah, so anyways, thanks for listening to my presentation, and hope you liked it. And that's it. Thank you.